Good evening. Welcome you all to the PSI online lecture series. This is the third year that we again start off with a new vision, with new series of lectures. And today, our lecture series is on cardiac action potentials and pacemakers. Uh, our presenter today is Dr. Vinay Chitturi, Associate Professor of Physiology, Department of Physiology, AIIMS Rajkot. He has his research interests in neurophysiology and digital signal processing. And with that, I welcome Dr. Vinay Chitturi and ask him to start his presentation.
Thank you, Dr.
Welcome,
Shanta.
Dr. Vinay. Yeah,
Mm, sure.
the stage
Uh,
is yours.
Thank you so much. I'll be starting my presentation. I'll share the window itself. Okay. Good morning, good evening, everyone, and welcome to a very fundamental topic in cardiac action potentials and pacemakers. So, as you can see, action potentials is a property we see in excitable cells and tissues. So, typically, action potentials would be seen in nerve fibers, skeletal muscle. Now, cardiac muscle is also a specialized form of muscle, and you see cardiac action potentials from the cardiac myocyte present at different levels of the heart. So if we see the heart itself, heart may primarily we have the atria and ventricle. So the cardiac muscle in them is called the atrial myocyte and the ventricular myocyte. So atrial muscle and ventricular muscle would have a specific form of action potential. Then similarly, there's also a specialized conductive tissue in the cardiac chambers. Nodal tissue and conductive pacemaker, conductive tissue also have their own kind of pacemakers. So pacemakers are capable of firing action potentials spontaneously. So through a property called automaticity. So a pacemaker action potential would be automatically produced at a regular rate, whereas cardiac muscle action potential is generated when cardiac action potential would be generated when pacemaker potentials propagate to the respective tissue. So if you'll see within a heart, we have certain pacemaker cells. So normally that would be the SA node, sinoatrial node. It's present in the posterior wall of the right ventricle. So pacemakers produce an automatic action potential. We call it a pacemaker. So they have certain ionic level membrane mechanisms which ensures that these pacemaker potentials, action potentials, are produced rhythmically over a period of time. So the SA node typically produces the rhythm of around 60 to 80, you know, action potentials per minute. Now, within cardiac muscle, you have the specific syncytial organization. So cardiac muscles are interconnected. So they have these things called intercalated discs. Gap junctions are present at intercalated discs. So electrical current can go from one cell to other across the intercalated disc. So typically what happens, pacemakers produce your action potential propagates along the length of cardiac muscle. Currents of this action potential can enter the neighboring cell through gap junctions and activate the process of production of action potential in these cells. So typically what happens in the heart, pacemakers, SA node starts your rhythm. The rhythm propagates across atria and ventricle, and atria and ventricle produce their corresponding action potential, and the entire heart beats at the rate of the dominant pacemaker. Right. So this is called a syncytial property of cardiac muscle. Pacemakers drive the entire heart through the cardiac conduction system. So now, what is this cardiac conduction system? Right, so typically when you see a pacemaker trained like this, pacemaker shuttered in the SA node, traveled action potentials are propagated through this cardiac conduction system, right? And as the impulse is propagated across the cardiac conduction system, action potentials are propagated. So there's some time taken for conduction, so that's why sometimes you see atria the first muscle to activate, then AV node, then bundle of His, then the ventricular fibers. So this specific sequence of spread of action potential across the heart gives rise to the typical waveforms we see in the ECG. Right, pacemaker cells are automatic, so remember the term automaticity. Right, they automatically produce action potentials. They do not have the contractile they have very few contractile actin myosin filament containing myofibrils. Whereas atrial and ventricular myocyte in the walls of the cardiac chambers, they are not automatic. They have to be triggered to fire by the pacemakers, but they contain your contractile myofibrils, which cause the contracting and pumping action of the heart. Right. So to quickly summarize the electrical conduction system, we have the SA node, sinoatrial node. We have the AV node at the AV junction, atrioventricular junction. We have interatrial pathways connecting the SA node to the AV node. They transmit. These are the rapid form of transmission from SA node to AV node. SA node also communicates across to the atrium, right? Then the bundle of His is produced from the AV node and it goes through the fibrous septum between atria and ventricle. So only electrical conduction between atria and ventricle through the bundle of His in normal condition. Right, bundle of His divides into its two component branches, the left bundle branch and the right bundle branch. Then the terminal fibers of the bundle of His and its branches are called the Purkinje fibers. So these are very rapid conducting. This entire system ensures the entire heart is electrically actuated in a very rapid sequence. Right? So you can see the conduction system in its full form. SA node, internodal pathways, AV node, bundle of His, left bundle branch, right bundle branch, then branches, fascicular branches of the bundles, and the terminal Purkinje fibers. Right? So the sequence of conduction gives rise to different kinds of action potentials. Now, if you'll see the morphology of the action potential, we have two primary morphologies. We have something called slow action potential and something called fast action potential. So slow and fast depends on the rate of rise of this upstroke of the action potential. Right? Not very obvious in this graph. So slow action potential seen in pacemaker cells. That would be SA node, AV node. Now, rapid action potentials seen in the myocyte, atrial myocyte, ventricular myocyte. Right? So if you can see the red ones, slow action potentials. The black ones, the black starred ones, fast action potentials. So fast action potentials have this sustained phase of depolarization called the plateau phase. So plateau phase, plateau phase, plateau phase, seen in fast action potential. Slow action potentials, slightly different morphology. Right? So what is the rate of this pacemakers? SA node, we say 60 to 80, can be changed by autonomic nervous system. AV node, 40 to 60. Purkinje fibers, roughly around 15 to 20. Now, typically, if there's normal conduction, these lower rate pacemakers would be overdriven by the faster pacemaker. So if the SA node is functioning normally, the cardiac conduction system is functioning normally, all action potentials produced in the heart, AV node, Purkinje fibers, muscle cells, they all will fire at the rate of SA node. So this is a phenomenon called overdrive suppression, where the SA node dominant pacemaker, fastest pacemaker, drives the rate of the all other parts of the heart. Right? So why is SA node the primary pacemaker? Fastest firing rate. And this action potential will transmit to other parts of the heart and drive it. So other pacemakers, so AV node which has capable of independent pacemaking would be suppressed by the higher rate of the SA node. This is a phenomenon called overdrive suppression. Right? So typically what happens? SA nodes automatically produces this pacemaker, this train of action potentials. This train of action potentials, each one would be conducted through the heart. So when it reaches the myocyte, you get a myocyte action potential. There'll be a small conduction delay between them, right? So what makes a pacemaker action potential unique? So we talk about intrinsic automaticity and rhythmicity. So this sequence of action potential changes, right, automatically take place in the pacemaker. It can take place independently. So if you take the heart outside and support it metabolically, you'll get this independent production of pacemakers without any external neural influences. And then there's no flat RMP, resting membrane potential. You can see this thing is not a flat line, it's slowly going up. Right? So let's try to understand the phases of this SA node pacemaker potential. Right? So these are conventions, phase 4. So I'll start with phase 4. So phase 4, basically it looks something like this. There's a slow rise, then there's a rapid rise, we call it phase 0. Then there's a fall, we call it phase 3. Then again phase 4, phase 3, phase 4, phase 0, phase 0. So remember the sequence: 0, 3, 4, or 4, 0, 3, 4, 0, 3. So there are three key phases: slow rise, slightly faster rise, then downstroke. Slow rise, upstroke, downstroke. So each of this is based on ionic currents across the membrane of the heart. So let's try to understand these currents. So typically pacemaker cells do not have a steady RMP. So cardiac muscle VCI has an RMP of minus 90 millivolts, right? So if it's not being stimulated externally, it will be a flat line membrane potential minus 90 millivolts. But pacemakers, they don't have a steady resting membrane potential. This is because of their ion channels. So typically at the end of the previous cycle, end of downstroke, the heart would be hyperpolarized. So typically it reaches a minus 70 membrane potential. Then because of intrinsic pacemaking currents, right, certain ion channels, right, this minus 70 slowly rises to a threshold of minus 40. Right, this phase is called the diastolic depolarization. Some people also call it a prepotential, right? So there's a slow rise from minus 70 to minus 40. We call this phase 4. So the pacemaking current. So there are certain ion channels which drive the pacemaking currents. I'll come back to it, right? So phase 4, IF, we call it the funny current. We'll come to why it's called the funny current. Now, once you hit this minus 40, right, minus 40 is the threshold for opening of calcium channels, T-type calcium channels. So once you go from minus 70 to minus 40, we open the calcium current, open the L-type and T-type calcium channels, and you get this calcium current because of opening of calcium channels. So this is a slightly slow upstroke compared to sodium, right? So when I say slow, it is slow in compared to the sodium channel, which is even faster. Hmm? So there's this depolarizing phase, we call it the upstroke. So calcium L-type current, right? Then once you depolarize, you reach a positive membrane voltage. Positive membrane voltage would activate the opening of voltage gated potassium channels. So potassium would go outwards now from inside to out. So it will cause repolarization. So this will come back to this thing. So now you get a more negative potential. We call this hyperpolarization, right? So just know the three phases: phase 4, funny current; phase 0, calcium L-type current; phase 3, potassium current. Now, if you'll see, what is the basis of this automaticity, right? So you saw the three phases, right? Slow depolarization, upstroke, downstroke, hyperpolarization. Now this hyperpolarization at the end of phase 3 activates your pacemaker current, right? So these channels are activated by hyperpolarization. So we call them HCN channels, hyperpolarization activated cyclic nucleated gated channels, right? So if you'll see, end of this thing, there's hyperpolarization. Hyperpolarization activates this channel. So that's why we call them HCN. Hyperpolarization activated, cyclic nucleated gated. Now, why do we call them funny current? So typically most ion channels are activated by depolarization. You saw calcium channel, depolarization based opening gating. Potassium channel, depolarization based opening. Now this is one atypical channel which is activated through hyperpolarization. So this ionic current activated by hyperpolarization. So that's why we call them funny currents. And there's a small component of what we call T-type calcium channels also to funny currents, right? So know your phases. Phase 4, funny current; phase 0, calcium current; phase 3, potassium current. So this occurs in a cyclical manner. So the activities of phase 4 activate calcium current. Phase 0, calcium current depolarization activates potassium current. Potassium current hyperpolarization activates phase 4. So it's a cycle. Phase 4 automatically leads to phase 0. Phase 0 automatically leads to phase 3. Phase 3 automatically leads to phase 4. So as long as the heart is functioning normally, this cycle keeps happening. So this entire process does not require external influences. It is intrinsic to the heart automatically, right? So automaticity. This happens automatically without any independent neural control, right? Now, that is pacemaker current. So if you see, cardiac muscle has a normal resting potential of minus 90 millivolts. We have this typical gradient: sodium high outside, potassium high inside, calcium high outside, right? Now, how is a ventricular myocyte action potential? So, ventricular myocyte is not automatic, right? So, unless it has an external stimulus, typically that external stimulus comes through the conduction system from one of the pacemakers. So, as long as there's no external stimulus connected to it from the neighboring cell, it will stay at RMP. What's the RMP? Minus 90 millivolts. Now, right? Now, once you, if you activate this pacemaker, it'll go through this sequence. So phase 0, phase 1, phase 2, phase 3, back to phase 4. So pacemakers, if you remember, do not have a 1 and 2. So why did this convention arise? So cardiac myocyte was studied first. So people gave them these names, phase 0, 1, 2, 3, 4. Then when phase cardiac pacemakers were studied, they didn't have a phase 1 and phase 2, but people stuck to the nomenclature. So cardiac pacemakers have phase 0, phase 3, phase 4, right? Right? Whereas cardiac myocyte has 0, 1, 2, 3, 4. So cardiac pacemakers do not have phase 1 and phase 2, right? So phase 0 is RMP. So the myocyte is happily sitting at RMP unless it is activated externally. Now, if and when the myocyte is activated externally, so typically there's a depolarizing current entering from the neighboring cells through the cardiac conduction system. So when this depolarizing current enters, right, there's opening of sodium voltage gated channels, right? Sodium. So pacemakers we had calcium voltage gated, here we have sodium voltage gated. Now sodium voltage can cause a rapid upstroke from minus 90 to plus 10, more than plus 10, right? This is much faster compared to calcium current. So this is why this is called fast action potential, right? Phase 0 sodium current, INA, right? So we have a sodium current. Now the membrane, another cell is depolarized. Now this at this thing, this voltages sodium channels inactivate, right? So there's no further sodium current. Now this voltage opens a group of channels called KTO, right? Potassium transient outward channels. So they give you this early repolarization phase, right? The small early repolarization. That's called phase 1, right? So phase 4, RMP. Phase 0, sodium current. Phase 1, potassium transient outwards. Now, what we have is opening of K channels, right? And calcium channels. So some calcium channels would have opened in this phase only, but most of them open in this phase for the sake of simplicity. Now, so we have this plateau phase where the depolarization is there and depolarization is maintained for a while. So typically we say around 200, 250 milliseconds. Depending on heart rate, it can be higher or lower. So this is called the plateau phase of cardiac action potential of myocyte. So plateau phase may we have sustained depolarization. Why is this happening? Both potassium and calcium channels are open. So any inward current to the calcium is almost balanced by outward current through the potassium. So if there's no net current flow, you maintain the potential. So for a potential difference, there has to be net current flow for a change in membrane potential. So phase 2, plateau phase, we have balanced currents, so membrane stays depolarized. Now what is the importance of this increased period of depolarization? I'll come to that. Now at the end of this calcium current is entering in is participating in EC coupling, so it goes into the sarcotubular system of the cardiac muscle. Calcium is further released from there. Calcium interacts with the myofilaments and this actual contraction, right? So phase 0, phase 1, phase 2 is the plateau phase. Now phase 3, the calcium channels closes, but potassium channels stay open. So there's more potassium flow outwards, right? So because there's potassium flow outwards from inside, you get this repolarization, right? Then potassium channels close and you get this RMP phase again, right? So just to quickly revise the currents, phase 4, resting membrane potentials. Phase 0, dominant current is sodium, little contribution of calcium too. Phase 1, potassium transient outwards. Phase 2, calcium and potassium currents balance each other, plateau phase. This is where the contraction takes place. Phase 3, potassium current. Phase 4, back to RMP. Right? So if you'll see, now if you see a nerve action potential, or a skeletal muscle action potential, it's in very few milliseconds. Nerve action potential is 1 to 2 milliseconds. Skeletal nerve action potential is 2 to 3 milliseconds. So it's possible for nerves and skeletal muscles to rapidly fire action potentials in a train of sequences. And this happens all the time when the muscles undergo normal technique contraction. Now, cardiac muscles, they have a longer action potential, right? 200 to 400 milliseconds depending on heart rate. So lower heart rates, higher duration, higher heart rates, lesser duration. So typically, if you'll see in a skeletal muscle, action potential has completely finished firing before a twitch contraction takes place. Cardiac muscle, uh, the contraction phase is coinciding with the action potential. And one very important thing is, when this membrane is depolarized, sodium channels stay inactivated. So as long as membrane potential doesn't come back to a more hyperpolarizing potential, sodium channels cannot be activated again. So what happens? No action potential can be fired by exciting the cardiac potential in this depolarized plateau phase. It has to come back somewhere down, right? So this is called the refractory period, when cardiac muscle cannot produce a second action potential. And what's happening? The duration of the action potential is so high, the muscle is almost relaxing by the time the action potential comes back down. So I cannot give a second action potential and lead to sustained tetanic contraction. So cardiac muscle cannot be tetanized. So skeletal muscle can be tetanized. You can maintain it in a constant state of contraction for quite some time. Cardiac muscle, it contracts, it relaxes, it contracts, it relaxes. And we want that, right? We want pumping. So the heart has to relax, fill with blood, contract, pump the blood. If it stays consistently contracted, pumping would not be efficient, right? So heart rate. So now these pacemakers, they have an intrinsic heart rate. I spoke about a SA node heart rate, intrinsic rate of 60 to 80. So this is an intrinsic property. So the pacemaker by itself, isolated heart would produce this rate. But as you know, heart rate can go up and down. So when you're exercising, heart rate goes up. Now when you're sleeping, heart rate typically goes down, right? So heart rate does change with body activity, neural influences, and body requirement. So how does this happen? So now there's an intrinsic pacemaker rhythm. So let's call this the SA node, right? So we have ourselves a phase 4, right? Phase 0, phase 3. So this is auto-rhythmic train of the intrinsic pacemaker rate. So let's say the intrinsic pacemaker rate is like this, right? Uh, Interaction potential duration of 1 second, right? So your typical heart rate would be 60 by this duration. So that would be a heart rate of 60, right? Now, what happens if I give parasympathetic stimulation? So, vagal stimulation. So, vagal stimulation would cause a decrease in heart rate. So, vagus acts on the muscarinic receptors of the heart, inhibitory in action, and they affect these ion channels. They affect the pacemaker ion channels, they affect the calcium channels, they affect the potassium channels. Now, the net effect of this action on these ion channels is some changes in the morphology of the action potential, right? So the newer train of action potentials, they'll be firing at a slower rate, so larger distance. So for example, if this is 1.5 seconds, your heart rate would be 40. Now, what are these key changes in the action potential? We can see them across all phases, right? There's decreased slope of the pacemaker potential. So go from minus 70 to minus 40, it takes a longer time, right? Then sometimes this hyperpolarization itself is higher. So instead of minus 70, if it goes to minus 80, it will take longer to go from minus 80 to minus 40. Then sometimes, because the calcium channels have changed, there's more positive threshold. So previously you were going from minus 70 to minus 40, right, in this one. But now you might be going from minus 40 to minus 30 at a slower slope. So this phase will take longer in time. Because this is taking longer in time, you're slowing down the entire cycle, you're decreasing the heart rate. So parasympathetic stimulation causes decreased pacemaker firing. That's one of its key effects on the heart. So we call this negative chronotropy. Chronos as in time. Now, what happens in sympathetic stimulation? Exact opposite effect as far as the pacemakers are concerned, right? Faster rate of firing of the pacemakers. So what's happening? Basically, the exact opposite of what happened in parasympathetic, right? Greater slope of the pacemaker potential, less negative hyperpolarization, less negative threshold. So you go from here to here faster, fire the action potentials at a faster rate, right? So, to quickly summarize my key findings, pacemaker potentials are automatic. So, to the right, we have ourselves a pacemaker potential, right? Pacemaker potential called slow potentials because it's upstroke slower compared to the fast upstroke of cardiac myocyte. Pacemaker potentials, no steady RMP, right? What they have is a diastolic depolarization, also called the pre-potential. This leads to automaticity. So this one leads to this one leads to this one leads to this. So you have this continuous rhythm of firing of action potentials depending on the ion channel properties, right? So let's quickly revisit. So the currents were responsible for them. Phase 4 pacemaker currents, funny current, HCN channels. Phase 0 voltage gated calcium channels, calcium current. Phase 3 potassium current through voltage gated potassium channels. Now cardiac myocyte, phase 4 is RMP, phase 0 is sodium channels, phase 1 potassium transient outputs, phase 2 calcium and potassium channels balance each other, plateau phase, long duration action potential. Phase 3 calcium currents, phase 4 is RMP again, right? Now what is the importance of knowing all these phases and knowing all these action potentials, knowing the channels, right? So we have arrhythmia. Now one of the mechanisms of arrhythmia, when we talk about arrhythmia, abnormal rhythms of the heart, there are two different key problems. So there could be what we call enhanced automaticity. So cells which are not supposed to automatically fire at faster rates than normal, or the other one is abnormal conduction. So, to address these issues of enhanced automaticity and abnormal conduction, you would want to target specific channels or involved in the cardiac action potentials, and the antiarrhythmic drugs which have been designed targeting the different phases of action potential and different ion channels mediating them. So, just a quick summary, as a build-up for pharmacology. So, there's a class 1 antiarrhythmic. They target the sodium channels which cause the upstroke. There's class 2 antiarrhythmics, beta blockers. They target the sympathetic system. Right? There's class 3 antiarrhythmics. They target the potassium channels of the heart. Class 4 antiarrhythmics, they challenge the calcium channels of the heart. So you get into the nitty gritty. There's lots of subtypes in each of them, and each of them is used as and when relevant for the treatment of different arrhythmias. So know your pacemaker potentials and know the cardiac conduction system. So our next sequence of cardiac physiology we'll get into the details of, so we saw the automaticity part, we can get into the conduction of the heart. So how does the conduction take place to the heart so the heart pumps cleanly? Um, so I thank our audience for.
Okay, thanks, Dr. Vinay. Such a wonderful lecture and with a wonderful presentation. I think we have some questions in the YouTube link. If you could check for the questions. If not, I
Um,
have. Are there any?
these are all
Uh,
comments. Good evening, good evening, good evening. Let me
no,
look
not
for the
that.
questions.
It's in the anyway. I have think a question that was cropping into my mind when you were going in through the cardiac action potentials. So, what actually happens during an acute myocardial infarction? Is it the action potentials that they are irregularly generated, or is it the pacemaker activity of the heart that is at default?
So myocardial infarction is a vascular disorder where there's compromised blood supply to the myocardium. So again, there's a full spectrum ranging from myocardial ischemia to myocardial infarction. Now, when you're using the term infarction, there's a very specific context. Infarction is death of cardiac muscle because of ischemia, right? Ischemic necrosis.
Okay.
So
Okay.
so, in the context of myocardial infarction, so any cells which are infarcted, right? So,
Yeah.
those cells are technically dead. So, that would cause a patch of myocardium which is not generating these action potentials,
Okay.
right? Now, in the
Okay.
context of myocardial ischemia, you get the full spectrum. So, erythemogenesis and abnormal conduction, injury currents, Injury currents are typically, we talk about injury currents from damaged part of the heart to other parts of the heart. But when we talk about this intracellular events taking place in the heart specifically, you get a full spectrum. So you can get everything from enhanced automaticity, you can get conduction failures, or you can get 100% failure. So it depends on the extent of disease. So if you are using the term infarction, then that tissue is dead. So there wouldn't be, yeah, that tissue wouldn't be the dead tissue would affect the conduction from neighboring cells too. But as
Okay.
far as the cellular levels are concerned, an infarcted cell is a dead cell.
Okay. Thank you. So we would like to thank Dr. Vinay Chitturi again for this wonderful lecture, and we hope to hear from you in the coming days as well.
Sure.
And thank you all.
Ma'am, I'll just let's play the outro.
Yeah.